If bacteria had a LinkedIn for unexpected career moves, Paracoccus sanguinis would probably top the list with a bio like, formerly thought to be an environmental microbe, now quietly thriving in human blood. Interests include immune modulation, redox balance, and not killing the host. It sounds almost fictional, a friendly bacterium calmly coexisting in what we were all taught is a sterile environment. Yet this recently studied microbe is part of a growing wave of discoveries reshaping our understanding of the bloodstream and its microbial entourage. First isolated from human blood samples during investigations into the blood microbiome, Paracoccus sanguinis belongs to a genus typically associated with soil, water, and extreme environments like salt flats and oxygen-poor zones. So, what's it doing inside us? Riding shotgun in a liquid highway packed with immune patrols? For starters, it seems to have developed some impressive survival skills. Unlike overt pathogens that trigger alarms and immune strikes, P. sanguinis appears to operate under the radar, possibly by avoiding classical immune recognition pathways or by maintaining such a low population density that it flies below inflammatory thresholds. It's the biological equivalent of a houseguest who tiptoes, does the dishes, and leaves no trace. This microbe's presence in healthy individuals has led to a radical shift in the sterile blood paradigm. It's not an infection in the classical sense. It doesn't cause fever, systemic inflammation, or organ damage. Instead, it seems to exist in a sort of peaceful symbiosis, possibly even offering benefits. Initial research shows that P. sanguinis produces metabolic byproducts that can influence host biology in subtle ways. One class of interest? Redox-active molecules that may help modulate oxidative stress. Oxidative stress is a foundational driver of aging and chronic disease, responsible for damaging DNA, proteins, and cell membranes. Some early studies suggest P. sanguinis may help maintain redox balance by producing enzymes like catalases or superoxide dismutase analogs, or by influencing host pathways like NRF2, which regulates antioxidant defenses. It's as if this bacterium showed up in your blood with a tiny fire extinguisher and said, "I got this." But it gets more interesting. P. sanguinis might have a role in immune tuning. While most bacteria in the bloodstream get a swift and aggressive immune response, this one seems to elicit mild or even tolerogenic reactions. It may stimulate regulatory T cells, TREGs, or dampen pro-inflammatory cytokines, which could be particularly relevant in the context of autoimmunity, aging, and chronic low-grade inflammation. It's like having a diplomatic attaché in your bloodstream, quietly encouraging immune cooperation instead of conflict. There are also hints that Paracoccus sanguinis may interact with endothelial cells, the lining of your blood vessels. These interactions could influence vascular tone, repair, or inflammation in microcirculation. If confirmed, this would put P. sanguinis in the same category as gut bacteria that influence cardiovascular risk through metabolites like TMAO, only with possibly positive effects. A microbe that keeps your vessels calm and your endothelium tight is one worth knowing, especially as we age and vascular health becomes a central concern. Of course, not everyone is ready to roll out the red carpet for this little bacterium. Critics argue that its detection may be due to contamination or DNA fragments from dead bacteria, not actual living residents. However, live culture experiments and microscopy have started to challenge that assumption, showing viable colonies and metabolic activity in blood samples. Still, it's early days. There's much we don't know about its life cycle, origin, and long-term stability in the body. Where does it come from? That's still an open mystery. One theory is that P. sanguinis translocates from the oral microbiome or gut, sneaking in through microscopic breaches in mucosal barriers. Another idea is that it's acquired environmentally and simply finds sanctuary in blood under specific conditions. Either way, its presence seems more tolerated than battled, suggesting that our immune system may recognize it as part of a broader, more nuanced ecosystem than previously understood. The idea that blood harbors friendly, or at least harmless bacteria is an unsettling but fascinating shift. If Paracoccus sanguinis is truly a commensal or mutualist, it could reshape how we approach blood diagnostics, immune therapy, and even aging interventions. Imagine future treatments that involve fostering beneficial blood microbes rather than eradicating all with antibiotics. Or a day when we sequence your bloodstream microbiome to detect imbalances before disease symptoms arise. It's speculative, sure, but not much more so than the now mainstream idea of gut-brain or gut-skin axis, which were once considered fringe. For now, Paracoccus sanguinis sits at the edge of mainstream microbiology, part curiosity, part potential collaborator. Whether it proves to be a guardian, a bystander, or something more complex remains to be seen. But one thing is clear: the bloodstream isn't the sterile desert we once imagined. It's a living, reactive, and possibly even cooperative ecosystem, and P. sanguinis might be one of its most polite residents.